Hi, my name is Jeanelle Boyer, and I am thrilled to be talking to you today about my research project for the last year or even more. And I'll be talking to you a little bit about FPIES and the gut microbiome. I've begun looking into this connection to see if there is a connection between the gut microbiome and the progression and the development of food protein-induced enterocolitis syndrome. So you might wonder, how did I come up with this idea? Why am I studying this connection between FPIES and the microbiome? And I am 100% inspired by my daughter. And here she is, at maybe 8 or 9 months old, her first food trial. She's attempting to eat carrots, which she loved, and unfortunately, they didn't work so well for her. It was a failed first food trial. But now 2 years later, she's eating carrots very successfully. So interestingly, I joke that I was sent this baby by the heavens because of my background in nutrition and my background in microbiology. Years ago, as I was working on my PhD at Cornell University, I was looking into the study of absorption of micronutrients. But down the road, I became interested in microbiology and began exploring the microbiome. I still remember my first conference, this microbial ecology conference, where I was introduced to the human microbiome, and I was absolutely fascinated about the implications for human health. So I joke that I was sent this baby because of my expertise in these two areas. So currently I'm a professor in health science at Keene State College, and long interest in the gut microbiome, but my new interest in FPIES is thanks to my daughter. I was passionate about the microbiome. I felt that it was very critical to human health and was pretty upset when I had to take antibiotics while pregnant, knowing that it would affect my microbiome. And I wondered if it would have any impact on my daughter. And then she was born by emergency C-section, which not planned, but another exposure to antibiotics. Despite all this antibiotic exposure, she was still a very happy, healthy baby girl. I think it was around 4 weeks old that she had her first cold. And after her first cold, she developed an ear infection. And again, I did not want to use antibiotics, but at the same time, I know ear infections and maybe a 5 or 6 week old at the time could be problematic. So along with her doctor, her physician's advice, we used antibiotics and treated the ear infection wonderfully. And I joke that after that she never had a normal diaper again. My happy, healthy baby was covered in eczema. She had mucousy stools with diarrhea, occasionally blood. She had severe reflux. She was diagnosed with GERD, Sandiford syndrome, where she would throw her head back and fling her arms up, almost looking like a neurological condition from the reflux. And I couldn't help but wonder if she was reacting to something I was eating, to food, that she was showing sensitivity to food. And it took a while to get a diagnosis. I think it was around 9 months old, and by that time I had limited my diet severely. I had tried a 5-food elimination diet in an effort to find something that she could thrive on. And I never had success with eliminating foods from my diet, but we luckily had a lot of success with Neocate. She thrived on her Neocate formula, and it took a couple of months, but eventually she passed broccoli as her first food, and she ate broccoli and all the related vegetables for about 6 months, and then all of a sudden started eating other foods. She's had numerous chronic reactions and 1 acute vomiting to shock reaction. But overall, I will say we've been very lucky in our journey with a lot of great support along the way. So she is my inspiration, and after my own personal story, I couldn't help but wonder if the microbiome was playing a role in the progression of her FPIES. And with my background in microbiology and my position at Keene State College, I feel very lucky that I have the opportunity to explore this connection in more detail. So before I go too far into this story, I want to say some thank yous. I couldn't have done this research without help from the FPIES Foundation and from a Keene State College faculty development grant. Both have supplied funds to support this current project. I'm also very grateful for Dr. Yuan from Mass General Hospital for providing support, for helping me discuss the clinical side of FPIES, for helping me develop surveys, and for discussing all these wonderful plans. So he's been a great collaborator. And I'm also very thankful for Microbiome, who's doing a lot of the science behind this project. And they actually are so excited and so supportive about this project that they have decided that they will fund my next project, which I'm extremely grateful for. All right, so first of all, I just want to spend some time talking about the human microbiome. What is the human microbiome? I know some of you may be new to this term, human microbiome. And the human microbiome refers to all of the bacteria in or on a human being. And it's estimated that there are 100 trillion bacteria on a human being, in and on us. That's 10 times more bacteria in or on us than actually human cells. So some could say that we are actually more bacterial than we are human. At the very least, we can be described as an ecosystem, a very complex ecosystem that's teeming with other life than ourselves. This is actually a picture of microbes, of the bacteria in a person's mouth. And my other fun stat is that there's more bacteria in your mouth than there are people that have ever lived on this planet. So billions of bacteria just in your mouth alone. To make it even more interesting, all of these bacteria have their own unique genome, which means there are way more bacterial genes in us than there are our own genes. And what does this mean for human health? Well, in some ways we are just beginning to learn, but the consensus is that these bacteria that live on or in us really do play a very critical role in human health. And to live a healthy lifestyle means to take care of all of the microbes on or in us. So we've had an interesting journey with microbes throughout the years. Currently, we have concern that the human microbiome is at risk, that we're losing diversity, that we don't have as many microbes, and we don't have the same microbes as we did 1, 2, or even 3 generations ago. And much of this is from some really valuable work over the last 100 years in combating infectious disease. So instead of embracing our microbiome, our little bacteria that live in or on us, most people think of bacteria as something like this guy or these guys, these kind of scary monsters, these germs that are just lurking in every corner, just waiting to make us sick. And this kind of approach to bacteria has been helpful in some ways. If we go back about 100 years ago, even just 50 years ago, we saw that some of the leading causes of death were infectious disease. And over the last 50 years, we've seen death from infectious disease decreasing. So there very much is a place for this approach to keeping the germs away and washing hands and taking antibiotics. It has helped significantly in reducing risk from infectious disease. However, I think a lot of times this happens in science and in medicine. We go to one extreme and then we realize where we really need to be is somewhere in the middle. So what we've seen with the decrease in infectious disease, we have seen an increase in inflammatory disease and autoimmune disease, and disease of a dysfunction of the immune system. And a lot of researchers are hypothesizing that this could in fact be due to damage to the human microbiome. And in an effort to eradicate infectious disease, we may have had some unintended consequences on the poor little microbes that are part of our human ecosystem. So this has led to this wonderful advance into the study of the human microbiome to try to really understand how these microbes that live on or in us can influence our health and wellness. And what we have learned is that for sure, this is complex. Each individual has a unique microbiome, and within each individual, we have, in a sense, different microbiomes. So, for example, the skin microbiome is primarily made up of actinobacteria in most people. Whereas if you take a look at something like, let's see, the colon or the gut microbiome, which I'll be focusing on, they're showing a lot of these firmicutes, which are seen in red, a lot of bacteroides, which are seen in purple. Whereas in the stomach, it's a lot of proteobacteria. So within the human, there are, in a sense, almost like these different microbiomes. And this varies between each person. So one person might have more firmicutes in the gut, another person might have more bacteroides, another person might have more actinobacteria than the average person. So this isn't going to be an easy picture to kind of elucidate. There's a lot of variability within a person and between people, and it's made it challenging to do this research. However, I would say that it is becoming more and more clear that this microbiome, that a healthy microbiome, really is critical to health. We know that the microbiome changes over our lifetime. Again, the colored graphs are just showing the relative abundance of different bacteria, and the big picture is just to see that it's changing over time. Infants and even the prenatal microbiome is getting a lot of attention these days. It is believed that there's this critical period before birth, that begins before birth, and then during the first 6 months of a baby's life, where the immune system starts to learn what is an invader and what's not an invader. That there's some programming that happens with the immune system. And evidence is suggesting that it's very closely tied to the microbes that are on or in an infant. And some evidence is starting to suggest that if the microbiome is not healthy, is not balanced correctly, it might have these long-term immunological impacts. It might affect a baby's immune system, so that as an adult or even as a child later in life, the immune system doesn't do as good of a job recognizing what's an invader and what's not an invader. And when you have immune systems that are overreacting, we start to see more autoimmune disease, more allergic disease, more inflammatory disease. So science has started to show that there are many impacts of the microbiome. For years we've known that the microbes in the gut play a big role in breaking down nutrients. They play a big role in digestion. However, things like impact on this development of the immune system, on resistance, on protecting against epithelial injury, these are some newer concepts. And I really want to focus on this development and training of the immune system point, because the immune system, for one, plays a key role in allergic disease, which is what I am, of course, most interested in. But the immune system affects almost every organ in the body. And if immune systems are getting trained differently than they maybe were 50 years ago, because of unusual microbiomes, different bacteria inhabiting the gut, it can have system-wide impacts. And sure enough, researchers are looking at the connection between the microbiome and autoimmune disease and allergic disease. They're looking at connections between the microbiome and health, mental health, anxiety, depression, cancer, heart disease. I mean, almost every organ system may be impacted by the human microbiome. But of course, my interest is primarily in allergy and allergic disease. So there has been also an explosion in the literature that supports this idea that the gut microbiome, as well as the skin microbiome, even the lung microbiome, might actually impact allergic disease. And evidence is suggesting that it probably starts before babies are even born. I've seen some interesting studies recently suggesting that even if mom spends more time on a farm, they're less likely to have babies with allergic disease. So this idea that exposure to a diverse array of microbes can be beneficial for a developing immune system. So a number of studies are starting to look at this connection between the microbiome and the progression of allergic disease. I've noted a couple on this slide. In an older study from about 7 years ago, they looked at the gut microbiome of infants, very young, at 1 week old, and then I think again at 2 months old. And they found that in these infants, if they had fewer of these lactobacillus and fewer of these bifidobacterium, they were then more likely to go on and develop food allergy. So babies with more lactobacillus and more bifidobacteria were less likely to develop food allergy. So this seems to kind of corroborate with this idea that early infant microbiome could potentially affect inflammatory disease down the road. I know some researchers are looking at connections between diabetes and other autoimmune disease. In a more recent study, I found this one interesting. The researchers looked at the gut microbiome of children with IgE allergies, like the hives, anaphylaxis, that kind of an allergy, compared to the non-IgE food allergy, which of course FPIES would fall into. And they did not look at FPIES specifically. However, they found that babies who develop food allergy did have a unique microbiome compared to babies that did not develop food allergy. In fact, they found 20 different types, 20 different genera of bacteria were different between this food allergy group and the non-food allergy group. And in addition, they found that the babies who had the IgE food allergy had a unique microbiome compared to even the babies that had non-IgE food allergy. And to be clear, the methods behind looking at the microbiome, it's as simple as a stool sample. Retrieving a stool sample, extracting the DNA from the stool sample, and then matching it to a database to get an idea of what bacteria are actually present in that stool sample. Now, over the years, so there are a number of studies that show microbiomes differ between babies who develop asthma, babies who don't develop asthma, and a number of other allergic diseases. However, there really hasn't been much or any research into this FPIES, food protein-induced enterocolitis syndrome. So I, with my background in microbiology and my baby who was allergic to everything, I'm very excited to start to explore this connection and perhaps find a difference, if there is a difference, that could explain anything related to perhaps the development of FPIES and offshoots of this that could lead into potential treatment, prevention, if we do find that there's a difference between the gut microbiome in the FPIES babies versus the non-allergic babies. Okay, and I should add that just because there's a difference in the microbiome, it doesn't always necessarily mean that the microbiome is causing the disease. So correlation does not always equal causation. However, more and more evidence is accumulating that the microbes probably do play a role in the development of allergic disease. And a couple of kind of well-known studies in the world of microbiome and food allergy involved a bacterium called Lactobacillus rhamnosus, sometimes known as Lactobacillus GG, and Clostridium butyricum, a type of Clostridial species. And in one study, they found that if babies were supplemented with Lactobacillus rhamnosus, which is a common microbe, it's found in a lot of probiotics. I believe it is, thanks to studies like this, it's starting to get more and more attention as a probiotic. They found that babies who had Lactobacillus rhamnosus supplementation were able to tolerate milk more quickly than babies who did not receive the Lactobacillus rhamnosus supplementation. And I should add too that initially in their study too, they found that the microbiome sure enough did differ between the allergic and the non-allergic babies. Lactobacillus rhamnosus doesn't necessarily colonize the gut well. However, they found that the presence of this bacterium seemed to shift the microbial population in the gut towards a population of bacteria that were anti-inflammatory. There's bacteria that produce butyrate, and butyrate is this short-chain fatty acid that tends to have anti-inflammatory effects. So it's thought that could be one of the ways that this Lactobacillus rhamnosus is helping build tolerance to milk in this study. The Clostridial species, so this was a study done in mice, and a couple studies leading up to it, they started to key in on this bacterium, this Clostridial bacterium. And if you hear Clostridia, you might think Clostridium difficile, which is a well-known pathogen. However, other types of Clostridia seem to be very beneficial. Clostridium butyricum is one of those butyrate producers that produces some anti-inflammatory compounds in the gut. And in this study, they found that in mice, Clostridium butyricum actually induced this compound called interleukin-22, IL-22. And when this compound is increased, it makes the intestines less permeable, less leaky, you could say. So that peanut allergen would have less of a chance of ever reaching the blood. And that, sure enough, these Clostridial species were protective in helping to prevent peanut allergy. So these two bacteria are getting a lot of attention, and just for so these, I think there's a lot of good work here, but the gut microbiome is very complex. So this is just the tip of the iceberg, I feel. There are literally hundreds of species of bacteria living in someone's gut. So there's a potential for a lot of more work here. Right, so scientists, researchers are starting to key in on some of the factors that could affect a gut microbiome. And I keep focusing on gut, but the entire human microbiome, and microbes are found all over. And the area, one of the areas that's getting a lot of attention are these early life factors. So they're finding that the way a baby was born, whether C-section or vaginal birth, can affect the microbiome. If born by C-section, of course the babies don't go through the birth canal. They don't pick up all of the mother's microbes that are inhabiting the birth canal. Instead, they might pick up more bacteria from the hospital, from skin contact. So it creates a unique microbiome. Breastfeeding has been found to be beneficial for microbiomes in that there's certain oligosaccharides, certain sugars in breast milk that feed some of these beneficial bacteria quite nicely. Even things like early skin-to-skin contact will affect this sharing of microbes for the baby. Now, it used to be the old thought is that the fetus, that before birth, that the baby was sterile, that there were no microbes that would affect the fetus. However, more and more evidence is suggesting that no, there are microbes that are present even before birth. And more interest is accumulating in the maternal microbiome, the mother's microbiome, that this may actually impact the health of the baby, potentially because of these shared microbes, but potentially because the immune system, that infant immune system, is already developing before birth. So the mother's microbial health may impact the baby's microbial health. The environment that you live in, I've seen a number of studies that suggest if you live in more rural environments and live a farming lifestyle, there's lower risk for allergy. That if you're connected, if you're out there in the dirt and more exposure to the outdoors, that's thought to be beneficial for the microbes. Stress has an interesting effect on the microbiome, and the microbiome has interesting impact on stress. So studies are suggesting that there are certain microbes that are associated with stress and associated with depression. And there's some early research that's even suggesting that some probiotic supplementation may actually help benefit those with stress and depression. Again, early work. In some mice models, they took microbes from stressed out mice, and they took microbes from non-stressed out mice, and they transferred the microbes to germ-free mice that were bred in these sterile conditions to not have a microbiome. And if you took the microbes from the stressed out mice and gave them to the germ-free mice, they became stressed out mice. If you took the relaxed germs, the relaxed microbes, and put them in the germ-free mice, the mice were more relaxed. So stress probably impacts the microbiome. The microbiome seems to impact stress as well. In diet, that old saying, you are what you eat. And I joke with my microbiology classes that you're not only what you eat, but you're also what your microbes eat. And when you're feeding yourself, you're feeding your microbes. And there's a lot of evidence now that certain foods are better for the microbe. And we'll get to that. Hygiene. This is a case where overly hygienic is probably not good for the microbiome. Now, hygiene, of course, is important in combating infectious disease. So I think we're entering this new age where we have to find a balance between helping our microbes and still preventing infectious disease. Because going back 100 years with no hygiene, less hygiene would not be good for the world of infectious disease. But I do believe we've gone too far with antimicrobial compounds in everything from dish soap, hand soap, to children's toys and clothing. That over-hygiene is damaging to our microbiome. For years, people have been talking about the overuse of antibiotics, that's contributing to lower microbial diversity. Research is suggesting that exercise is good for microbiome, the gut microbiome, the human microbiome, sleep, and then there's this interesting and complex genetic component. There definitely seems to be an interplay between genetics and our microbes. And I think the field of epigenetics, this idea that our genetics, our genes, can be turned on or turned off, and thanks to our environment, will be an interesting field, knowing that there's probably a good chance that microbes are actually influencing our genetics, just as our genetics are creating an environment that's unique and unique for the microbes to live in. So it is impacting the microbes that live in or on us. I wanted to make this point. I know talking with other mothers of FPIES babies, when they start to hear that the maternal microbiome may impact their baby or early life factors, they start to feel responsible for FPIES. And I just want to be very clear that this is a much bigger problem than an individual. And I am confident that every mother out there is doing the best that they can moment to moment for their child. So there's no need to beat oneself up. But we live in a culture where people don't have access to the good food that supports a healthy microbiome in many situations. Plant-based diet, high-fiber diet, organic, pesticide-free. Literally, there are food deserts in this country where there is not access to this kind of food. And even if you have access, sometimes it is not accessible due to cost. So our diet, which is, again, been slowly shifting over the years towards a diet that is probably not as good for our microbiome. We're also living in a culture where we don't have as much access to the outdoors. We live in environments that are much cleaner than we used to. And that alone could contribute to a different or a less diverse microbiome. And then there are some old medical practices, many of which have helped in this battle against infectious disease. But I would argue, again, potentially overdoing, that we're coming to a point where we need to control infectious disease, but we need to protect the microbiome. So things like overusing antibiotics, not necessarily having the appropriate testing to see if antibiotics are needed, if it's a viral illness or if it's a bacterial illness. And then meanwhile, we really live in such a culture of fear where there's scary monsters lurking around all the corners, and I think a lot of times those scary monsters are the germs or the microbes. So if we can start to shift our perspective that not all bacteria are bad, that many of them are good, many of them are beneficial, I believe we can start to change our culture where we're always afraid of illness, knowing that the more good microbes we have, the less at risk will be for infectious disease. A healthy microbiome tends to mean a healthier immune system. So to tie this into my FPIES project, you may be wondering, well, how are we going to study FPIES in the microbiome? And to this date, I've recruited about 80 families. And of these 80 families, about half were FPIES babies, FPIES infants, half were allergy-free infants. All of the infants in the study are 12 months or younger. We're hoping to target that early microbiome, but again, they had to be old enough to know that they had FPIES or not. So we're going 12 months or younger. And all were free of antibiotic usage for the past month before stool sampling. After I recruited these wonderful families who I'm very thankful for participating in this research, they were asked to complete a short survey, and in this survey we asked questions about what are the FPIES reactions like, how many triggers, but also some questions about factors that might influence the microbiome, such as antibiotic usage, exposure to pets, type of birth, C-section or vaginal birth. Then, in addition to the survey, stool samples were collected thanks to kits sent from Ubiome. So Ubiome has created a very easy, user-friendly method for sampling, in this case a diaper, and just getting a small amount of fecal material, putting it in a solution to keep the DNA stable, and then sending it back to Ubiome, where they'll be doing the sequencing to look at the different types of microbes that are found in these stool samples. So what we'll be looking at, we will be looking at the big question, is there a difference? Are the allergy-free babies, do they have a unique microbiome compared to the FPIES babies? And we'll look at this in different levels, at the phylum, all the way down to genus, maybe even some species details. If it does differ, then the big question is, how does it differ? Are there microbes that are more abundant in an FPIES infant? My hypothesis is that we might see some either pathogenic or even opportunistic pathogens or bacteria that are just generally more inflammatory. It wouldn't surprise me, and I'm hypothesizing that we'll see more of those in the FPIES infants compared to the controls, the allergy-free controls. We'll also be looking to see if anything is lacking. It wouldn't surprise me in some of these previous studies on IgE allergy, they're finding that certain bacteria aren't as abundant. So both of those, that would all be interesting information. And then I do want to see if there's lifestyle factors that are associated with FPIES, such as antibiotic usage, birth method, breastfeeding, or formula feeding, as these things could have an influence on the microbiome. And it could start to offer some more evidence to put together this picture of how our environment and the environment that we're born into can impact allergic disease. So where are we now? We have collected about 75 surveys. Most of the stool samples have been returned. We're still waiting on a couple more, and we're still awaiting sequencing on a couple more. And we are just starting the data analysis, and I'm predicting that, well, it'll take me several months to analyze the data, or more. It could be, there'll be a lot of data generated. But my hope is to have some solid results by the end of the year. Since I don't have the microbiome data to share with you, I wanted to at least share some of the survey data, because I think there's some interesting things from the survey. So first, a little bit about our FPIES infants. You'll notice, so one of the questions we asked, are they acute, are they chronic, or are they both acute and chronic? And let me back up and say that this is all self-reporting. So these are surveys that were sent online that families filled out. So these are not like clinical diagnoses straight from the physician. Though we did ask the participants that they only participate if they did have an official diagnosis from a physician. So acute FPIES is probably the better understood FPIES, the FPIES that comes with the acute vomiting 2 to 4 hours after food ingestion, sometimes leading to the point of shock. The chronic FPIES, I feel like, is still a little less clear and characterized by abdominal discomfort, diarrhea, chronic diarrhea, maybe chronic vomiting, not that severe acute vomiting that we see with the acute FPIES. And I think one of the challenges for those who are dealing with FPIES is that it's not a well understood disease at all, and the chronic FPIES is even less well understood. So I thought this was interesting when I look at these numbers within our group. 19.5% had acute FPIES, so 24% had chronic FPIES, and 56% are experiencing both chronic and acute. And I think this just suggests that it just supports this idea that there's a lot to learn about the chronic FPIES, but it's a worthy cause. It's common. There are a lot of babies who are experiencing these chronic symptoms. I wanted to share the age range. One of the questions I asked was, when did you first notice symptoms? And I thought it was interesting that most noticed symptoms, so almost 50%, 48.8% noticed symptoms at 0 to 2 months. A small group, 12%, noticed them at birth. And then you see there's another kind of spike, 24% at 5 to 6 months, which I'm guessing is probably around solid food introduction. However, I think this is relevant. If people are noticing symptoms between 0 to 2 months, there's an opportunity to start offering support and help as we start to increase our awareness and our understanding of FPIES. Now, at 0 to 2 months, babies, of course, are not on solid food. They're either breastfed or formula fed. And I know one of the challenges I experienced as an FPIES parent was I noticed symptoms before she had an acute reaction, but they were symptoms that were really hard. That were really hard for her, and it was really hard for me as a parent to watch. And most of these symptoms were through breast milk. And yet, when you read the FPIES literature, there really isn't a lot out there about reactions through breast milk. And I believe if we look, there's about 38% experience vomiting, but I think it's not so much that acute vomiting, to shock, but more of these chronic symptoms. But I think this also, again, offers an opportunity with this awareness to perhaps support and intervene and help families that might be struggling with some of these chronic symptoms. It might also be an opportunity then to be cautious with solid food introduction and not to necessarily not introduce foods, because babies can experience these symptoms without FPIES. But perhaps, I know in my own situation, I didn't start with any of the common FPIES triggers. So I started with the yellow and green vegetables, and I think because of that, I delayed those acute FPIES reactions. My own daughter did have an acute FPIES reaction, but not until 14 months when we were trialing turkey. And it was much less severe than other families describe. So 34 out of 41 infants the families claim that they did experience the symptoms with breast milk. And I should say these numbers are percentages of the 34 that experience the symptoms to breast milk. And again, what I see most commonly, about 70% experience diarrhea with mucus or blood, about 70% more night wakings, close to 80% acid reflux, so the number experiencing vomiting, quite a few, 55% experiencing a skin rash, and then the changes in mood or temperament, close to 60%. Again, when I look at we looked at general symptoms to the solid foods, not just to breast milk or to formula. And we see, again, a wide variety of symptoms, the most common being vomiting to the food, but quite a few of these chronic symptoms as well. 78% with acid reflux, 70% vomiting to the point of lethargy, and again, diarrhea without mucus or blood, diarrhea with mucus or blood, 56% and 73%, and the increased night waking. So we're seeing a whole array of symptoms in our population. And some of the most interesting data that I wanted to make sure I presented today are the differences between our FPIES patients, the FPIES participants, and the non-FPIES participants. And I've highlighted in red the two categories where we saw a significant difference. We did not see a significant difference between formula feeding. We did not see a significant difference between rate of cesarean section. This first column are the FPIES infants, the second column are the allergy-free infants. Interestingly, there are actually more C-sections in the allergy-free infants. However, it is not reaching statistical significance, so p-value of 0.3, so not considered significant. Antibiotic usage by mother before pregnancy, no significant difference. However, the FPIES infants had more antibiotic usage by the mother during pregnancy, and that did reach statistical significance. So 48.8% of mothers of FPIES infants used antibiotics while pregnant, and only 23.5% of the allergy-free infants had mothers that used antibiotics. So I think that's relevant. I think that would be interesting to look into, and it kind of supports this idea that the maternal microbiome does have an impact on the infant microbiome. Again, not a lot of difference between antibiotic usage during delivery or antibiotic usage by the mother while breastfeeding. It is higher in FPIES infants, but not statistically significant. However, antibiotic usage by the infant was significantly different in FPIES babies compared to the allergy-free babies, with a p-value of 0.036. So anything less than 0.05 is considered significant. So 43.9% of FPIES babies received antibiotics, and 20.59% of the allergy-free infants received antibiotics. So significantly more antibiotic usage in the FPIES infants. I didn't see a significant difference with families with a cat or a dog. And interestingly, so I looked at the sex. From what I've read, it seems to be more common in males, and we did see more males in our study group. 68% of our FPIES group were males compared to 52%. However, this did not reach significance, p-value of 0.16, getting closer to significance, but still not statistically significant. The highlights of our survey data, and I should add that we are just in the beginning of analyzing the data. I think I had my last survey was collected 2 days ago, so there's more I will probably be doing with this data. But already, I do think it's very interesting that there's more antibiotic usage in FPIES infants and in the mothers of FPIES infants, suggesting, again, that there could be an impact on the microbiome, and that this microbiome could be impacting the development of FPIES. I also think it's interesting that most families were noticing symptoms when they were very young, and that many noticed them through breast milk. I bring this up because I think this is an opportunity to increase awareness and to potentially help and support and to intervene and to reach out to these families, hopefully before they're experiencing the acute reactions and the more severe reactions that might come with solid food introduction. But I also know from experience that that was a stressful time for me. My infant had not yet experienced an acute reaction, and yet the chronic symptoms to breast milk were very challenging. And any kind of support that we can offer these families, I think, would be beneficial. I also thought it was interesting that most of the FPIES infants experience both chronic and acute reactions, which to me just kind of highlights this need to understand more about the chronic FPIES and to better define the chronic FPIES. So in the future, my goal is to complete the analysis of the microbiome data. I know that's a lot of the meat of this project, and I'm very excited to get into that analysis. I would also like to begin another project to continue to look at the microbiome, comparing older FPIES toddlers that have outgrown FPIES to FPIES toddlers and children that have not outgrown FPIES, hypothesizing that we would see a difference in the microbiome if the microbiome is at play, and then comparing them to allergy-free children, hypothesizing that those who outgrow FPIES might be more similar to the allergy-free children. And thanks to new funding from Microbiome, they've offered to pay for all of the stool sampling kits so that we can complete that project, which is very exciting. Long term, I would like to collect enough pilot data to get funding for a longitudinal, more long-term study following infants and their families to keep understanding the microbiome and the role that it might play in FPIES. I also have even longer-term hopes of, if we do see that the microbiome is playing a role, are there potentials for intervention to help prevent this dysbiosis, this unhealthy microbiome? Would things like probiotics help? Are there interventions that could help after FPIES has developed? And if we know that it's the microbiome, if we know that the microbiome plays a role, again, I don't think that it's 100% microbiome, but I'm hypothesizing that the microbiome plays a role. It could allow for an avenue of research and exploration to help with prevention and intervention. All right, so to end, I just wanted to end with my recommendation. So if after listening to this talk you think, oh, I want to be kind to my microbiome, I want to cultivate the ecosystem of microbes within me, I ended with my list of ways to make friends with your microbiome. So one of the best things you can do is get outside and connect with nature. Years ago I came across a study that looked at even depression, and we found that folks who garden were less likely to be depressed, and I think it was due to an exposure to certain microbes in the soil. Get outside, and at the very least even open your windows and let the microbes in. So this is time to embrace the microbes. The more healthy microbes you have, the less space there is for the bad guys, for the pathogens. Get dirty and avoid over-cleanliness. Try to avoid overuse of hand sanitizer and antibacterial soaps. Interestingly, the FDA has just ruled to pull triclosan from the antibacterial soap. So I actually think that was a good move to help with our microbiomes. And lastly, an exercise has been shown to benefit the microbiome. So I share with you a picture of a good friends of mine. He's doing everything he can for his microbiome. He's just done a cyclocross race. He's covered in dirt. He's exercising, and then he is so kindly sharing his microbiome with the love of his life as well. So we get to share our microbes with others. All right. There's some evidence that exposure to animals, pets, might be beneficial for a microbiome. Again, some early research looking at even just being on a farm, around farm animals, might help prevent allergy. So I've used that as an excuse to make sure we have a dog, lease a pony, and visit animals as much as we possibly can. So part of my own therapy for my daughter is to spend time on farms, which I love as an animal lover. Again, using antibiotics only when medically necessary, and I think again, part of this is going to be a shift in how we approach infectious disease. Antibiotics are critical, they're absolutely critical for certain infectious disease, and without them, again, we don't want to go back 50 years where infectious disease was a greater killer. However, I do believe they are still overused, so finding that balance where we can start to kind of come up with methods and ways to determine when are these antibiotics truly needed, and using them appropriately. Relaxation, so meditation, has been found to be beneficial for the microbiome. And then of course diet, so you are what your microbes eat, and there's a lot of evidence that you can rapidly change your microbiome through diet. I know some big studies showing that in humans who ate an animal-based diet, when they switched to a plant-based diet, within 10 days they saw a shift in the microbiome, a beneficial shift in the microbiome. So my recommendations that I put here are more for the average person who may already have or I should say who doesn't have a dysbiosis, who's not having extreme gut issues. In general, it's good to eat a plant-based diet, a high-fiber diet. Try to avoid the pesticides. Try to go organic, so that we are eliminating some of those compounds that aren't so good for our little microbes that live in or on us. Fermented foods, if they're raw fermented foods, they have live microorganisms in them, which can be beneficial. And avoiding the highly processed and highly preserved foods. kind of the traditional Western diet these days, it tends to be higher and higher in these processed and preserved foods, and they are less good for our microbiome. Now, I put a question mark after probiotics, and I guess in a sense, especially for the FPIES babies, I probably could have put a question mark behind a lot of these. I do think that this is a very individual, if a person is having problems with their gut microbiome, that this could actually be a very, it could require an individualized approach. And we still need a lot more work and research. However, some people with severe dysbiosis may not be able to tolerate a high fiber diet. So then what do we do? If someone has histamine issues, the fermented foods may not be appropriate. Probiotics, I said there's some evidence that L. rhamnosus or Lactobacillus GG and Clostridium butyricum are effective for helping prevent allergy. Though I still think there's a lot of work to be done in this area. There's still a lot that we don't know. What are the effects of the probiotics? Are they effective? Are they not effective? So I think there's evidence that is starting to gather that is showing that probiotics definitely have potential to be helpful. And I keep hoping for more research to keep supporting that and to help developing and learning more about the microbiome so that we even know what are the bacteria that we need and what role do they play in our health and wellness. So lastly, I just want to say thank you. I'll end with a big smile from my daughter. This is part of her watermelon trial this summer, and I'm happy to say she has passed it and happily eats watermelon on most days. That's one of her favorite foods. So I'll end with a big smile, and I just want to say thank you for listening. And I wish you all the best, especially the families who are out there on their FPIES journey. If you would like to contact me, Keene State College, you can look for my name, Jeanelle Boyer at Keene State College, and I am happy to talk more about FPIES and the microbiome. Thank you.